Hello dear student. Today we will start with our second lecture of protein metabolism. In first lecture of protein metabolism, we have studied dynamic state of our body protein, importance of amino acid pool, phases of protein metabolism, and then we studied the digestion and absorption of protein. So let's share my screen. So today we are going to discuss two important metabolic processes regarding metabolism of protein, that is the transamination and deamination reactions of amino acid. In last lecture we had seen that the amino acid what we get in the blood, they mostly we get as a result of the digestion and absorption from dietary protein and degradation of the body protein. So as we all know that this amino acids, most part of it is utilized for synthesis of new structural protein and synthesis of non-protein nitrogenous compound. So this comes under the anabolism part of the protein metabolism. Now let's go to the catabolism part of the amino acid metabolism or protein metabolism. So what we see that the first step in the catabolism of any amino acid is the removal of amino group. The removal of amino group is known as the deamination process, and this because of removal of this amino group, the resultant compound which is formed is known as a carbon skeleton, which is mostly the keto acid. But now we know that we have 20 standard amino acids which are present in our body, which take part in the metabolism. But here in this process there is one restriction, that not all amino acid can undergo deamination. So, and apart from this, only glutamine or glutamic acid is the important amino acid, because that has capacity to freely undergo deamination to produce ammonium ion, and which are utilized for the production of the urea. So what can be the remedy when we want all amino acid to undergo deamination? So here is the rescue. The process is known as transamination. Now, what is a transamination? By its literal meaning, it says that it is a transfer of amino group. So it is a transfer of amino group from amino acid to the keto acid, and which will lead to the formation of new pair of keto acid and amino acid. So this transamination process help us to concentrate all amino group of amino acid to form glutamate. And how it is, you can see the when the keto acid alpha ketoglutarate takes part in the transamination reaction, the all amino acid loads their amino group to alpha ketoglutarate, and we get resultant product, that is the glutamate amino acid. And we have previously discussed that this glutamate is very important. Even do you remember that in amino acid pool 50% of quantity is of this glutamate. So this glutamate has a capacity to undergo deamination. So you can see by transamination reaction, all amino acids are channelized to the glutamate, and this glutamate undergo deamination. Deamination means removal of free ammonia, and this free ammonia, as it is toxic, it will be detoxified to form urea in liver, and this urea is excreted in the urine. So this is the catabolic part of the amino acid. Now what is the fate of keto acid which is formed here? Now keto acid can undergo complete oxidation to produce energy in the form of ATP. It can take part in the gluconeogenesis and form glucose. Even it is a precursor for synthesis of the fat, and through the transamination reaction again it can form non-essential amino acid. So this diagrams gives you overview of catabolism of amino acid, and here we learned that the transamination and deamination are very important process for every amino acid to undergo catabolism. So let's discuss this transamination and deamination reaction in today's lecture. So as the name suggests, this transamination is the transfer of amino acid from it is a transfer of amino group from amino acid to the keto acid to form new amino acid and new keto acid. So it is clearly seen that it is only shuffling of amino group from one amino acid to the keto acid to form new amino acid and new keto acid. Now this reaction occurs in the cytosol and mitochondria of every cell, but it is occur in more quantity in liver, kidney, brain and heart. And there is a restriction, like only 3 keto acid can take part in the transamination reaction. The one major keto acid we know, we have discussed in last slide, that is the alpha ketoglutarate, which will give us glutamate as a product. So first one is alpha ketoglutarate. Apart from alpha ketoglutarate, pyruvic acid and oxaloacetic acid can take part in the transamination reaction. Now, let's come to the enzyme. The enzyme which catalyzes this reaction is known as transaminases or aminotransferase. Now, it's important, this transaminase need one coenzyme, and that is a pyridoxal phosphate or PLP, which is a derivative of vitamin B6. Now, let's see why this transaminase need pyridoxal phosphate. So in this diagram, you can see here, like, this is the amino acid, and this pyridoxal phosphate, it is linked with the transaminase enzyme. So this pyridoxal phosphate accept the amino group from the amino acid and it hold it for some time to form pyridoxamine phosphate, and it releases keto acid. Now this pyridoxamine phosphate, it gives its amino group to another keto acid like alpha ketoglutaric acid, and there is a formation of glutamic acid and pyridoxal phosphate is regenerated. So this pyridoxal phosphate, it acts like a ship's base here, because it holds amino group of this amino acid for some time, and then it donates to the another keto acid. And this mechanism is also known as a ping pong or bye-bye mechanism of transamination reaction. So now, as all amino acid, not all amino acid undergoes transamination reaction. Exception is lysine, threonine, proline, and some books also mention hydroxyproline. So these amino acid cannot undergo transamination reaction. So what are the importance of transamination reaction? Few points we have already noted down in last slide. So let's revise it. So significance is it helps in the interconversion of amino acid which are in short supply. Every amino acid, non-essential amino acid, can be synthesized from another amino acid. So this is the significance of transamination reaction. Then second significance is very important, that it channel all amino acid to the glutamate and to some extent in the aspartate. And we know that glutamate is only amino acid which undergo deamination. Now, third important significance is it is a transamination reaction connects between carbohydrate and protein metabolism. We know that in TCA cycle there are keto acids. This keto acid undergo transamination to form different amino acids. So it is a connection between carbohydrate and protein metabolism. Let's see now examples of transaminases. So let's study two transaminases which are more oftenly used in clinical practice, and in your theory it will help also. So this is you can see this is the AST. So AST is an aspartate transaminase. Previously it was known as a SGOT, that is serum glutamate oxaloacetate transaminase. AST is a new name and it is derived from the donor amino acid. As clearly you can see here, aspartate aspartate donates its amino group to alpha-ketoglutarate to form glutamate, and it forms keto acid, that is the oxaloacetate. So this is the aspartate transaminase. Again, it will require pyridoxal phosphate as a coenzyme. Now let's take example of second transaminases, which is used very commonly, that is ALT. So ALT, it stands for alanine. So it is alanine transfer its amino group to alpha-ketoglutarate to form a pyruvate and glutamate. And here the enzyme is known as alanine transferase. In previously it was known as the serum glutamate pyruvate transaminase. Now, why these two transaminase we are discussing? Because these two transaminase have the clinical significance. Like AST or SGOT, it is used as a marker for myocardial tissue, and its level increases much high in case of myocardial infarction. Even its level increases in liver disorder, muscular dystrophies, and myositis. While this ALT or SGPT, it acts as a marker enzyme for hepatic tissue, and the level increases very high in hepatocellular damage, as in case of acute hepatitis, even drug-induced hepatitis. So these were the transaminases of clinical importance. Most oftenly, the short note is asked in the transamination alone for 3 marks, or even you can get long question on this both processes, like transamination and deamination. Now let's move to the another important reaction, that is a deamination. At its meaning says that it is the D, means removal of amino group. Yes, so this process involves elimination of amino group from amino acid to free ammonia. So this is the deamination. Here you can see the amino acid is removed and this amino acid is converted into keto acids. Now in our body there are many type of deamination occurs, but predominantly we are going to study two type of deamination, that is the oxidative deamination and non-oxidative deamination. In oxidative deamination there is a liberation of ammonia with there is a liberation of ammonia from amino acid and it is coupled with the oxidation. While in non-oxidative deamination, there is a deamination but without undergoing oxidation. Now let's discuss what is the oxidative deamination. As we have discussed, it is a deamination with the coupled with oxidation. So most of this oxidative deamination occurs in liver and kidney, and the importance is that it provides ammonia for the urea synthesis, and it also provides keto acids which can be used in different reaction. We have discussed already in first slide. Now the enzymes which cause this oxidative deamination are two main type of enzyme. One is the glutamate dehydrogenase, which can be concerned as a main enzyme, and second is the amino acid oxidases. So let's see one by one. So glutamate dehydrogenase is a major enzyme for oxidative deamination. Mostly it is the mitochondrial enzyme and it is zinc dependent enzyme. Now you can see this is a one-step reaction. Like glutamate, in presence of glutamate dehydrogenase removes ammonium ion and converts this glutamate into alpha-ketoglutarate. But this enzyme needs coenzyme. The coenzyme is, it can use the specialty of this enzyme is it can use NAD or NADP both. So now, though you can see this is the first step, single step reaction, but this is not a single step reaction. You can see what happened. So here glutamic acid in first step, it would be oxidized to alpha aminoglutaric acid, and this is intermediate. Suddenly this intermediate alpha aminoglutaric acid will lose its ammonia, or it gives ammonia and it is converted into alpha ketoglutarate acid. Now what is the speciality of this reaction? As you can see, this is a reversible reaction, and the rate of reaction can be shifted to either side depending on the availability of substrate and availability of the ATP. So here you can see it is importance of this is it is a reversible reaction, occurs in mitochondria. It can use NAD or NADP both as a coenzyme. Aminoglutarate is intermediate, we have seen. Now, how it is regulated. So here you can see, after the protein-rich diet, our blood is full of glutamate. Why? Because at this availability of glutamate will inhibit this glutamate dehydrogenase. Even if the cell energy levels are high, like if there is a more amount of ATP or GTP, it will also inhibit this enzyme glutamate dehydrogenase. On the contrary, if energy levels are low, like ATPs are less in the cell, this level, it would be stimulated. There would be more conversion of glutamate into alpha ketoglutarate, and this alpha ketoglutarate will be utilized as a source of energy in TCA cycle. Then steroids and thyroxine, these are the these hormones. It inhibits this alpha ketoglutarate dehydrogenase. Now let's discuss the another enzyme, that is the amino acid oxidases. We all know that in our body we have L-amino acids. So L-amino acid oxidizes this L-amino acid and it converts it into the keto acids with the liberation of free ammonia. Now this L-amino acid oxidases is flavoprotein and it is a zinc dependent enzyme. Again, it can use FAD and FMN both. So in oxidation, this FAD is converted into FADH2 while FMN is converted into FMNH2. Now in this process of conversion of FMNH2 to FMN again, there is a synthesis, H2O2 is released and this H2O2 is neutralized by enzyme catalase. Now if we compare with the L-amino efficiency of L-amino acid oxidases with the D-amino acid oxidases, its concentration is low. Moreover, glycine and dicarboxylic acid cannot be acted upon by this enzyme. So the role of L-amino acid oxidases in the oxidative deamination is significantly low as compared to D-amino acid oxidases. Now, as we all know that D-amino acid does not present in the mammalian tissue, but they are present in food and vegetable and even microorganism. And in our body, we get this D-amino acid through our food. So how it is metabolized? You can see this D-amino acid, there is an enzyme D-amino acid oxidases. It will act on D-amino acid and this D-amino acid is converted into alpha keto acid. Now once this alpha keto acid is formed, it undergoes transamination and through this transamination reaction there is a formation of L-amino acid. Now this L-amino acid is it can take part in any of the metabolic reaction. It can undergo gluconeogenesis, it can give you energy, or it can even go through the formation of fat. So that's why the amino acid levels are high in our body, and it helps in the formation of L-amino acids. So significance of D-amino acid oxidases, it convert unnatural amino acid into L-amino acid, which can undergo various processes. So here we have ending with the discussion of all the three enzymes which are responsible for oxidative deamination. Now let's discuss the non-oxidative deamination. As we discussed earlier, this non-oxidative deamination is deamination without oxidation, and most of the time you can see it takes out ammonium ion from the amino acid. So here you can see the histidase is an enzyme which causes non-oxidative deamination of histidine, and it converts it into the urocanic acid. And this enzyme also need pyridoxal phosphate for its activity. Now, cysteine is a sulfur-containing amino acid. An enzyme which causes non-oxidative deamination of cysteine is known as desulfhydrase. So, in first step, this desulfhydrase removes sulfhydryl H2S from the cysteine and convert it into the amino acid. Now, these amino acid can be easily deaminated to form the pyruvic acid. Now coming another three amino acid, they are the hydroxyl group containing amino acids like serine, threonine, and homoserine. These undergo non-oxidative deamination with single enzyme, that is a dehydratase. Dehydrase. So these dehydrase removes water and ammonium ions. First, it removes water to convert this amino acid into amino acid, and this amino acid gives ammonia. It releases ammonia and get converted into pyruvic acid. So these are the enzymes which are responsible for non-oxidative deamination. Now, this slide shows that for catabolism of any amino acid, deamination is required, and this removal of ammonia from the amino acid, it takes couple of reaction. Like you can see that every cell transaminase is an enzyme which is present in the cytosol of almost every cell, and this amino acid, by the process of transamination, all ammonia is concentrated on the glutamate. Here, glutamate is acting as a uh, acceptor for amino group. So this glutamate is transported to the liver, and in liver it undergo oxidative deamination, and this oxidative deamination, it releases ammonia from the this glutamate, and it is used for the synthesis of urea, and this urea is exhaled. So here by this diagram we can see that though this transamination and oxidative deamination occurring at different organ, but it's still they though they are physically they are occurring at different places, but physiologically they are joined together, they're coupled together. That's why this both reactions are also termed together as a transdeamination. So transdeamination is an important part in the catabolism of amino acid, and this ammonia which is released by this reaction, it is toxic. So it needs to be handled in a very proper way, and this we will see in next lecture, that is the fate of ammonia. So now we will stop here, and we will continue in next class regarding the fate of ammonia. Thank you everyone. If you like this video, please hit the like button and you can put your questions in the comment box. Thank you so much.